Dear students, welcome to tomorrow online classes. So today we will start with the next cell organelle. So that is very important cell organelle. It is ribosomes, right? So these ribosomes are commonly known as the protein factories, right? So why these are called as protein factories is because they help in synthesis of proteins. What are these ribosomes, how their structure is, and what are the functions of these ribosomes? All these are the things let us study now itself, right? So let us start with the ribosomes. So these ribosomes are nothing but they are the submicroscopic naked ribonucleoprotein granular structures. These are commonly known as, right? What these are? Ribosomes are. These are submicroscopic naked nucleoproteins these are. So let us see, these are submicroscopic naked in the sense that these are already these are membraneless cell organelles, right? These are membraneless cell organelles, hence these are called as naked. Why these are called as ribonucleoproteins? The question arises. These are called as ribonucleoproteins. Why? Because these are made up of rRNA as well as proteins. Hence these are called as ribonucleoproteins. What these are commonly known as? Ribonucleoproteins. rRNA, these are made up of rRNA as well as proteins. Hence these are called as ribonucleoproteins. Right? And so let us see about the discovery of these ribosomes. These ribosomes were first discovered by Porter. Porter is the name of the scientist, the one who saw these granular structures present in the cytoplasm, and these are named as ribosomes. Okay, so Porter is the discoverer of these ribosomes, and later Talade saw the same ribosomes as granular structures in the cytoplasm of the animal cell. He also saw in animal cell. Porter also saw in animal cell, and he named them as Talade granules. What these are known as? Talade granules. Okay, and so later Robinson and Brown are the two scientists, the one who saw the same Talade granules of the ribosomes in plant cell. They saw the same granular structures in plant cells, right? So Robinson and Brown are the two scientists. So this is about the little bit discovery of these protein factories, right? Why these are called as protein factories? These are called as protein factories. Why these are commonly known as protein factories? Because they help in the synthesis of the proteins. These help in synthesis of proteins. hence called as protein factors. And these are also known as what? So these are also known as cell organelles within the cell organelles. Cell organelles within cell organelles. These are described as cell organelles within the cell organelles. Why? Because these are present in, right? These are present in cell organelles like mitochondria. Where they are present? They are present in mitochondria. They are present in chloroplasts. They are also present in chloroplasts. So, and these are also present within the nucleus. These are also present within the nucleus. So, nucleus, chloroplast, mitochondria, these are nothing but they are cell organelles. So within these cell organelles, if these protein factors called as ribosomes, they are present, then what they are described as? So they are described as the cell organelles within the cell organelles. Then where these occur actually? Occurrence of these granular structures, that is pallid granules of the ribosomes. So these are occurring in all kinds of cells. That is nothing but prokaryotic cell as well as eukaryotic cell. In all kinds of cells, these are present in. Right? So these are present in both the kinds of, that is prokaryotic cells as well as eukaryotic cells. They are present both in prokaryotic cells as well as eukaryotic cells. Right? These are occurring in. And these are also occurring in mitochondria, chloroplast as well as nucleus. Right? So such kind of cell organelles, these are also available over here. And the number, whenever you consider the number of ribosomes, so these are maximum in the cytoplasm of the cells. So they are more in number when compared to all other cell organelles. These are more in number. So they are largest number of cell organelles, that is ribosomes, are present in the plant cell as well as animal cell, right? And so these number of these ribosomes, that is more in those cells which are synthesizing more amount of proteins. So these are more in number in cells which synthesize more proteins. In those cells only these are maximum in number, right? So we came to know what these ribosomes are, why they are called as ribonucleoproteins, why they are called as naked one, and the discovery about these ribosomes, and why they are called as protein factories, why they are named as cell organelles within the cell organelles, and their respective occurrence as well as number that we discussed now itself. So let us start with the structure of these ribosomes. How the structure of these ribosomes, let us see now. Let us see the ultrastructure of this ribosome, right? Ultra structure of ribosome. What is ultrastructure of ribosome? Ultrastructure in the sense that the structure which is seen under the electron microscope is called as ultrastructure. Okay. The structure of ribosome seen under electron microscope called as ultrastructure of ribosome. Whenever we see this ribosome, ribosome has two major subunits. Two subunits are there in each ribosome. What are those subunits? So one is larger subunit and another one is smaller subunit. There are two subunits that we come across in the ribosomes. One is larger subunit and another one is smaller subunit. As you are seeing here, so this is smaller subunit and larger subunit. These are the two subunits that we come across over here, and this subunit, smaller subunit, has, so the middle cleft is there, cleft, and this is called as platform, this is called as head. So these are the three parts that we come across in the smaller subunit. One is middle cleft, and this is the head of the ribosomal subunit, and this is the platform of the ribosomal subunit. And so there you are seeing here the central protuberance. The central protuberance is seen over here. This is called as the stalk of the stalk of the larger subunit, and this is called as ridge. So these are the three structures that we come across over here in larger subunit. So these are looking to be as so complementary to each other. They can fix each other, right? So these two, I mean, this is smaller subunit, this is smaller subunit is fixed over the larger subunit, right? Larger subunit. Just like a cap which is fixed on the larger subunit. That way, both of themselves fix each other and they looks to be as complementary to each other. They are meant especially for fixing each other. And in what way? Both of themselves fuse each other, right? So there is a need of Mg++ ion for fusion of these two subunits, right? So there is a need of magnesium, so +2, that is plus ions for fusion of both ribosomal subunits. So when there is Mg++ ion is there, that helps in union of these two ribosomes, right? Ribosomal subunit. I shall show you here very simply. This is one ribosomal subunit and this is another ribosomal subunit. So this is smaller subunit, this is larger subunit, right? Larger subunit. Both of themselves fuse each other in presence of Mg plus ion over here, right? And what is the concentration of this Mg++ is needed for the fusion of both the subunits? So it is about 0.001 m. Mg++ ion is needed. This much of concentration of Mg++ ion is needed for the fusion of both the subunits over here, right? So 0.001 n, right? That much of Mg+ ion is needed for the fusion of both the. If the concentration increased about 10 times more than this one, so 10 times more concentration of Mg++ ion leads to formation of ribosomal dimers. So leads to formation of ribosomal dimers over here. How the ribosomal dimers are formed over here? Let us see here. So what is this one? This is larger subunit and this is smaller subunit. Okay, this is one ribosome. Okay, and this is another ribosome. This is another ribosome. Okay, so these are the two ribosomes. Whatever it may be, 70S ribosome, 70S ribosome. So if concentration is 10 times more, then so these two fuse each other forming the dimer. See here, this is one ribosomal subunit, this is another ribosomal subunit, and this is another subunit, and this is another subunit. See here, both of them fuse each other, forming the 100S ribosomal dimer. What it is called as ribosomal dimer. It is called as ribosomal dimer. It is called as. In the same way, if ribosomal subunits are, that is nothing but this ribosome is ATS. Usually this is present in prokaryotic cell, and this is present in eukaryotic cell. These are fused, ATS and ribosome, both are fused, then it forms the structure like this. And it forms 120 ribosomal subunit. So ATS together form 120S, 70S together form 100S ribosomal dimers. It forms as a result of the increased concentration of what? Increased concentration of magnesium ion, which is needed for the fusion of both the ribosomal subunits. Instead of fusion of only the two subunits of one ribosome, there two ribosomes fuse each other when the concentration is 10 times increased over here, right? So this is the fact what we must know. Now let us see the types of ribosomes now. See here, there are different types of ribosomes. So the ribosomes present in the prokaryotic cell ribosomes, and eukaryotic cell ribosomes. So the ribosomes present in the prokaryotic cell are usually 70S ribosomes. These are usually 80S ribosomes are present over here. So this is the fact that is known to us. See here, in prokaryotic cell, the ribosomal subunits, they are made up of, chemically made up of 60% of rRNA is there and 40% of proteins. What they are consisting of? 60% of rRNA is there, 40% of proteins is there. In this, it is reverse. 40% of rRNA is there, and here 60% of proteins are there. So this is the chemical composition of both the types of ribosomes over here. So I shall draw the diagram of prokaryotic as well as eukaryotic ribosomal subunits. See here, this is prokaryotic, that is 70s ribosome, and this is 80s ribosome. 80s ribosome. So S stands for Swedberg's unit. It is standing for Swedberg unit. I have already explained regarding this one in the previous classes. So see here, in both the subunits, that is 70s ribosome, it is 50 years subunit and it is 30 years subunit. This is smaller subunit. This is smaller subunit. This is larger subunit. Right? And this is also smaller subunit. And this is larger subunit. Chemically, what these two subunits are made up of? So let us see. Here it is 40s ribosomal subunit. Here it is 60s ribosomal subunit. Total it is 80s. Total it is 70s ribosomal subunits. See here, chemically what these two subunits of the ribosomes made up of. Let us see that one very clearly. See here, so this is 70s ribosomal subunit. You can observe here 70s ribosome, and this is 80s ribosome. 80s ribosome. Chemically, these two are made up of, already we know. So these two are made up of proteins as well as rRNA. Then what are the strands of rRNA that are present in this subunit and in this subunit? So here 16 years strand of rRNA is present over here. rRNA is present over here in this subunit, smaller subunit of prokaryotic cell. And here, so there are two, that is about 23 years as well as 5 years. 5 years strands of rRNA that we come across over here. So in this larger subunit of prokaryotic cell, prokaryotic RNA, prokaryotic ribosome. Okay, so these are the type of rRNAs that are present in both the subunits. So larger subunit as well as smaller subunit. In smaller subunit, 16s strand of rRNA is there. In larger subunit, so two are there. One is 23 years and another one is 5 years. Next one. In 80s ribosomal subunit, so there the rRNAs are somewhat different. See here, it is 18 years strand of rRNA is present in the smaller subunit, and in larger subunit it is 28 years. Remember here 3 that we come across over here. So 28 years plus 5 years plus 5.8 years are the 3 ribosomal strands that we come across. That is nothing but 3 rRNA strands that we come across over here. Ribosomal RNA. rRNA means ribosomal RNA. So this is the RNA which forms the structure of ribosomes. This is very clear to you, I hope so. So right. So this is how the chemical composition of these, so 2 types of ribosomes is very clear now. Okay. This is 70s, this is 80s ribosomal subunits. So this is one fact. And one more thing, another type of ribosomes that we come across in prokaryotes. In prokaryotes, there we come across one type of ribosomes that is nothing but polysome or polyribosomes. Polyribosomes or polysomes that we see in prokaryotic cells. So say for example in bacteria, already I have told you about this one. So what is this polysome? Very simple structure. See here, this is one ribosome and this is another ribosome. This is another ribosome. This is another ribosome. This is another ribosome. Such kind of many ribosomes are joined together forming an elongated chain of ribosomes over here. Right? And all of these are binded with the help of a single mRNA strand. What is this one? mRNA strand is there. mRNA strand is there. So that binds all the ribosomes over here. Chain of ribosomes. So these are the ribosomes that we come across. Okay. These are the ribosomes. Chain of ribosomes that we come across. See here. Okay, so ribosomes are usually called as what? Protein factories. So proteins are synthesized according to the genetical information what is present in mRNA. So this is nothing but mRNA strand. So there is one genetical information is there for the synthesis of one particular protein in mRNA. According to that genetical information, the type of protein is synthesized by these ribosomes. Is it very clear? See here. These larger subunits of these ribosomes, they are attached to endoplasmic reticulum. Outer surface of the endoplasmic reticulum, they are attached. And so these smaller subunits, they are binded to the mRNA. They are binded to mRNA. They are binded to mRNA, right? So I hope it is very clear. So now both the subunits bind each other, right? And these larger subunits leave a tunnel. They leave a tunnel into or an opening into the lumen of endoplasmic reticulum. So they leave one tunnel, one small hole, so or the opening into the endoplasmic reticulum. See here, suppose this is endoplasmic reticulum. Okay. To this endoplasmic reticulum, larger subunit is attached, and it leaves a small opening over here. This small opening itself is called as a tunnel, right? So this is smaller subunit. Think that is smaller subunit. This is smaller subunit. This is larger subunit, which is binded to what? It is binded to the outer surface of the endoplasmic reticulum and leaves a small opening over here. And this small opening is called as what? Tunnel. What we call it as commonly. So tunnel. Right? Why it leaves? The proteins what are synthesized over here. So those proteins are left into the lumen of this endoplasmic reticulum, and those proteins are circulated across the cell. Already I have told you endoplasmic reticulum acts as the circulatory system in the cell. right? So it helps in circulation of all the proteins which are synthesized by these bounded ribosomes. These are called. Already you know, free ribosomes are there, bounded ribosomes are there. There are different types of ribosomes. Free ribosomes are sailing freely in the cytoplasm, and bounded ribosomes are attached to the outer surface of the nucleus, and they are also attached to the outer surface of the endoplasmic reticulum, right? So like this they are called as bounded ribosomes. Okay. This tunnel is left for the collection of the proteins which are synthesized by the ribosomes. I hope it is very clear to you now, right? So this is how the whole system is there. In endoplasmic reticulum, one of the functions we wrote, since they are giving the surface for the attachment of the ribosomes on their surface, it becomes a rough endoplasmic reticulum, and it also associates with the ribosomes for the synthesis of proteins I had mentioned there. So because of this reason only. Okay, so now it is very clear how the attachment of these ribosomes that take place to the outer surface and leaves a tunnel. And the attachment that take place with the help of a specialized protein called as ribofourin. Ribofourin 1 and 2. Ribofourin 1 and 2 are the specialized proteins that help in binding of this ribosome to the outer surface of the endoplasmic reticulum. Okay, so this is also fact over. So there are two types of ribosomes that we come across. You write those two types of ribosomes now. Those two types, based on attachment of these ribosomes, they are not attached to any structures, right? Based on that, there are types of ribosomes. One is called as bounded ribosomes is one, and second one is called as free ribosomes. Bounded ribosomes and free ribosomes. Bounded ribosomes, they are attached to or bounded to the endoplasmic reticulum, and they are simply bounded to the inner membrane. So inner membrane or the membrane of the prokaryotic cell rarely, and they are free ribosomes in case of prokaryotes. Okay, these are bounded ribosomes. Free ribosomes are sailing freely in. So they are sailing freely in the cytoplasm. Free ribosomes they are called as, and bounded ribosomes, free ribosomes are the two types of ribosomes that we come across here. And the next topic is very important one, biogenesis. Biogenesis of ribosomes. What is this biogenesis of ribosomes? That is nothing but synthesis of ribosomes. Where does it take place? How does it take place? Let us see where does it take place now. Right? So biogenesis, free ribosomes in prokaryotes that takes place in the cytoplasm only. That takes place in cytoplasm only. But bounded ribosomes, see here, bounded ribosomes are also here. So these ribosomes, especially bounded ribosomes as well as free ribosomes, bounded ribosomes are also here. So if it is nucleus, Inside the nucleus there is another naked cell organelle is there, and that another naked cell organelle, so which is membraneless, and it is called as nucleolus. What it is known as? Nucleolus. In this nucleolus, what happens is synthesis of these ribosomes take place. What take place? These ribosomes take place. Synthesis of ribosomes take place inside the membraneless cell organelle, that is nucleolus. Inside the nucleolus, ribosomes take place. Hence, nucleolus is called as ribosomal factory. Ribosomal factory is called as. What it is known as? Ribosomal factory it is called as. Why? Because synthesis of ribosomes that take place inside the nucleolus. And later these ribosomes they come into the nucleoplasm, and ultimately they come out of this nucleus through nuclear pores. See here, nuclear pores are here, and through these nuclear pores they come out into the cytoplasm. See here, cytoplasm. It is. Into the cytoplasm they come out, right, and perform protein synthesis in the cytoplasm. Cytoplasm is the site where the ribosomes are present and synthesize the proteins. Okay, I hope it is very clear to you now itself, right? So the ribosomal factories are the nucleolus, and from the nucleolus it comes to the nucleoplasm. I mean, ribosomes come into the nucleoplasm, and from the nucleoplasm through the nuclear pores they come out of the nucleus into the cytoplasm, where the protein synthesis starts. I hope it is very clear about this one, right? Biogenesis of these ribosomes. Okay, so this is one more thing. And so some more information about these ribosomes are proper folding of these protein molecules and transportation of these protein molecules that takes place with the help of a specialized proteins. See here, already I had shown you in the diagram. So the ribosomes are attached to what? Endoplasmic reticulum with the help of riboforin 1 and 2 proteins, and they synthesize the proteins and produce those proteins, and they take that one into the lumen of the endoplasmic reticulum. There the folding of those amino acids chain that take place, folding and refolding that take place, and transport of the same proteins that take place. So with the help of a specialized protein called as chaperone. Is the name of the protein which help in folding and transport of these proteins synthesized by the ribosomes. So this is all about the ultrastructure of a ribosome, and what are the functions of a ribosome? Let us see now. Functions number 1. So these ribosomes already you know they are called as protein factories. So means they help in synthesis of proteins. They help in the synthesis of proteins is one thing. And second one is, so these ribosomes help in attachment of different types of RNAs, right? So they give surface for the attachment of these RNAs like So mRNA as well as tRNA. This is also another function of this one. Third one, very simplest one. So these ribosomes provide certain enzymes as well as factors for synthesis of polypeptides. These are nothing but proteins. Polypeptides means proteins itself, right? So this is also one more function of what? These ribosomes. By this we finished one more cell organelle, that is ribosome, right? And the next one is cytoskeletal system. Very important one, that is cytoskeletal system. So this is nothing but the internal framework of the eukaryotic cell. Basically in prokaryotic cell this endoskeletal system is not present over here, or the cytoskeletal system is not present in prokaryotic cell. It is an internal framework of eukaryotic cells. It is absent in prokaryotes. It is absent in prokaryotic cells. So, and this is made up of many tubules and filaments of some structural proteins. Some of the structural proteins it is made up of, right? And this endoskeletal system of the cell means, imagine yourself, the whole body of the human being itself is the cell. Inside this cell there is a bone system, that is nothing but endoskeletal system. So it gives particular shape for the cell, and it gives mechanical support to the cell, and it also helps in movement of the cell even, right? Imagine yourself, that kind of endoskeletal system is usually seen in case of cells also, and it is made up of many number of filamentous proteinaceous structures, and it is made up of many tubular proteinaceous structures. And what are those? Let us see. And this endoskeletal system that extend from the nuclear membrane up to the plasma membrane of the cell. It extends from nuclear membrane up to plasma membrane of cell itself. See here, one more point to be noted over here, how it binds to the plasma membrane. So very important one, vinculin is the protein that binds the endoskeletal system, all these tubular or filamentous structure, to the inner membrane itself, right? So vinculin is the name of the protein that binds the endoskeletal system to the plasma membrane, right? So this is also very important point to be noted. So let us see. So what are the structures that form the endoskeletal system of the cell now? Keith Porter is the name of the scientist, the one who discovered this one with the assistance of all his assistants, right? Keith Porter is the name of the scientist, the one who identified this one, discovered this one, and this endoskeletal system or cytoskeletal system, is made up of three structural proteins. What are those structures that form the cytoskeletal system? First one is microtubules. Second one is microfilaments, and third one is intermediate filaments. Are the three structures that form this cytoskeletal system of the cell. Microtubules are made up of the protein tubulin. Very important points these are. It is made up of tubulin, and microfilaments are made up of the actin, the protein called as actin, and intermediate filaments. These are made up of different types of proteins. So it depends on the type of cell in which they are, right? So tubulin, actin, and different types of proteins, they form microtubules, microfilaments, intermediate filaments respectively. Okay. So these microtubules, they are in diameter, they are about 25 nanometer in diameter, and actin, so they form microfilaments, they are about 7 mm, that is very thin one. So acting as a thread-like structures which bind these microtubules as well as intermediate filaments, and these are extending 8 to 12 nanometer in diameter. So what they are? Intermediate filaments. So these are the three types of structural proteins that form the cytoskeletal system inside the cell. And so all these three, microtubules, microfilaments, intermediate filaments, all these three are binded with the help of trabecular strands. So they are respectively binded with trabecular strands respectively. So this is how the endoskeletal system of a cell is made up of. So Keith Porter is the name of the scientist, the one who discovered this one. Okay, it is made up of microtubules, microfilaments as well as intermediate filaments. So all these are the things you understand yourself. Now we finished the cytoskeletal system as well as ribosomes are the kind of the cell organelles, and let us go through the next cell organelle in the next video. Thank you.